Neurocritical conditions, which are basically accidents of your brain, have high death rates or poor outcomes despite the changing times and medical advancements. They have many differences in their types, but all of them have a common feature, which is secondary brain damage by inflammation, oxidation, and excitotoxicity. This research will focus on one such pathway responsible for secondary brain damage: arginine metabolism. Arginine, a nitrogen rich amino acid, is a substrate for nitric oxide, and nitric oxide is responsible for local vascular autoregulation in the vessels of our body. This production reaction is mediated by nitric oxide synthase. Additionally, arginine can be methylated to dimethylarginine, DMA, which has inhibitory effects on nitric oxide synthase. Firstly, we will look into intracellular hemorrhage. Several studies have shown a decrease in arginine and its metabolites in the blood of ICH patients. It was especially significant in vegetative cases, while the ones who recovered had an initial higher levels of arginine. One possibility is that decreased level of arginine means less production of NO, thus vasodilation would not occur and there would be more damage in ICH cases. Subarachnoid hemorrhage is associated with terrible vasospasms, and they raise the comorbidity and mortality of the patients. A strong correlation of DMP was found in high comorbid patients of SAH. An associated increase in CRP and IL-6, which are inflammatory proteins, and a strong increase in gap junction protein connexin 43 was also observed. DMP inhibits nitric oxide synthase, thus, it might cause a decrease in NO production, which might again cause a loss of vasodilation and brain damage. Role of arginine metabolism in TBI is complex. Several studies have shown a decrease in nitric oxide with paradoxical improvement in condition of patients. NO causes vasodilation, and the vasoconstrictive effect of low NO limits the lesion in TBI. In genetically controlled rats, an increase in arginase in TBI caused mixed results due to loss of both vasoconstriction and vasodilation. CN105 and arginine rich peptide have shown improvement in mortality of ICH cases. Gap junction inhibitors decrease the levels of DMA and its response, thus have shown to benefit SAH cases. For TBI and approach inhibiting NO production have shown benefit.